Hello and welcome, everyone. Thank you so much for joining us for this fourth annual Black in Immuno Week, a week where we will celebrate, amplify, and support Black voices in immunology. Before we begin, we would like to thank our major sponsors who have made this week possible, and you can find our full list of sponsors on our website. Don't forget to tweet about our session today using dash AIML Immuno, with each word capitalized for accessibility purposes. We'll have a live caption and ASL interpreters that are with us today and during the whole session. And you can click on CC to have at the bottom of the screen to access the captions. You can submit questions through the comment function on the platform, and especially the comment function, not the Q&A. As a reminder, we require all audience members to be polite and respectful while in the Black Immuno space. If we see anything offensive, your comment will be deleted and immediately you'll be removed from the platform. After the conclusion of the week, recorded videos will be posted on YouTube on our channel and with all the transcripts that will be made available. You can find all this information on the website blackinimmuno.org. My name is Joel Babdor. I am an assistant professor at the University of Pennsylvania, and I'll be the moderator for this great session today. And I will start by introducing our two speakers that are eminent scientists and researchers in the field of AI, ML, machine learning, and artificial intelligence for a fantastic panel discussion where we will explore how AI/ML is being used in the field of immunology. So I'll start introducing our speakers, and I'll start with Danielle Belgrave. Danielle Belgrave is a distinguished professional in the biomedical industry in AI/ML, and in particular, she currently serves as the Vice President of AI/ML at GSK in London. With over a decade of experience, she has held key roles at DeepMind, Microsoft, and Imperial College London. Notably, her work at Imperial College London focused on applying statistical data science to understand disease heterogeneity, particularly in asthma and allergic diseases. Dr. Belgrave's dedication to merging scientific expertise with cutting-edge technology is all that we need for the future of immunology. Thank you for joining us, Danielle.
Thank you.
And now let me introduce Bryson, who is coming back to Black in Immuno Week for the second time, or maybe the third time. I think we've invited you multiple times because you're one of our favorite. Bryson is an associate professor of biological engineering at MIT and a core member of the Ragon Institute of MGH MIT at Harvard. Bryan completed a degree in mechanical engineering at MIT in 2007 and a PhD in biological engineering at MIT in 2013. After graduate school, Bryan went to the Harvard T.H. Chan School of Public Health, where he trained with Sarah Fortune and became interested in new approaches to examine host pathogen interactions in tuberculosis infection. Bryan joined the MIT as a faculty in 2018, and his lab works on infectious disease and developing new vaccines for TB. Thank you so much for joining us again, Bryan, and I'm really excited to hear your thoughts today.
Thanks for the invite.
All right, so let's start with, I guess, an easy question, but actually that might not be that easy. There's those terms, AI, ML, artificial intelligence, machine learning, are being used a lot in different contexts. It is kind of a buzzword today. Actually, the definition of it is not necessarily as clear as we would think. So can we, for the public to really have a good sense of what we're talking about, can we please define artificial intelligence, machine learning, and how those terms are, used together and why? Danielle, you want to start?
Start with the hard question. Okay, so I think if we think of artificial intelligence, we can think of machine learning as a subset of artificial intelligence. So artificial intelligence is all about creating systems and creating systems of intelligence that are not human intelligence, but learn from some sort of human substrate. So we can think of data. So a lot of us work with data, and we are creating intelligent systems through data. So we can think of different types of artificial intelligence systems. We can think of general systems that can solve many problems across different domains. So in science, in gaming, and these systems are translatable across different domains. I think that ChatGPT we can think of as one of the newest sort of intelligent systems that goes across several different domains and can extract knowledge from information and from data. So that's how I think of artificial intelligence. Machine learning is a subset of artificial intelligence, and machine learning is more of a mathematical, statistical, and computer science discipline. And it's based on data. So how can we extract information from data? How can we extract knowledge from data? So machine learning is a data-driven approach. So sometimes we use that data to generate hypotheses or to test hypotheses. So the difference, so my initial sort of background is in statistics, and we can think of machine learning as a way of scaling that to build models that learn from data and that are in a way autonomous. So systems that learn automatically, similar to artificial intelligence as well. So I'm curious to hear what Bryan has to say on this as well.
So it's really interesting because I'm actually teaching like a machine learning module in my class right now. And I actually share the definitions that Danielle offered. Oftentimes the way that I start telling the students is like machine learning is about letting the machine learn a set of rules without giving it explicit instruction, right? We're telling it, here's a set of perhaps input-output relationships in biology, but we don't actually know why the cancer cells died with this drug. We just have high throughput transcriptional data in all these different conditions. And now what you want the machine to do is learn the rules without you saying, well, it has to be this gene that drives this, right? It's about learning those patterns kind of in a systematic way, using these principles of mathematics and computer science. So I 100% am aligned on that.
Fantastic. Thank you both for defining these terms and the concepts that are behind these terms. What you just said, Bryan, kind of bring in mind the idea of the black box part of AI. And could you both comment a little bit on that? The fact that we don't fully understand how the algorithm is treating the data and well, there are ways that we can figure out what's happening, but we can't really fully understand what the machine learning approach is doing. Can you both comment a little bit on that black box?
Yeah, so this is really interesting because especially in biological settings, we value very much mechanism oftentimes. Mechanism is how we develop new drugs, new targets, et cetera, et cetera. And I think the thing that I'll say here is that I tend to be of the viewpoint that different types of machine learning approaches are more or less black box. And so I do think that for example, if I do use techniques that are based in kind of regression-based approaches, then I actually really do get, coefficients that correlate a particular biological measurement to an output. And so that gives me some way to weight a particular input to a particular biological output. By contrast, everybody's like deep learning. I think a challenge can be sometimes, how do I extract mechanistic insight from these really deep learning models, but I think people are trying that. And I think the other thing to consider is what do you want your model to tell you, right? Are you hoping, to borrow something that Danielle offered earlier, are you using this to potentially nominate new hypotheses? In which case you really want to be able to name a biologic hypothesis, right? I would like to say, this molecule, this protein contributes to host cell death, right? And so you want to use that in that way. By contrast, you can see lots of examples of deep learning models in, for example, medical imaging, which allow for early detection of tumors. Do you really need to know that why you were able to successfully early determine and spot a tumor? So I think when we think about the black box nature of it, is how much are we willing to accept the black box? Because I think for different types of biological applications, one might be willing to accept the black box because, like, hey, it let me do early tumor detection. I don't know how it worked, right? Versus, I'm trying to understand why this small molecule I developed killed all my cells. Well, maybe there you would really like to know mechanistically what the target is, and you need a different type of machine learning model for that.
Yeah, I love that comment and the way that you're, breaking down, situations where we want to know and situations where we don't necessarily need to know, right? If we can save patients by using the right drug because the machine learning approach is telling us to do so, yeah, we don't need to know why. If it's working, that's what's important for the clinician, I guess. That makes a lot of sense. Danielle, do you want to add on that?
Yeah, I agree with what Bryan said. I think that the term black box is very nuanced, and it depends on who the end user is of such systems. And I think that one of the latent factors in everyone's mind when we think about black boxes is around interpretability. And we worry about black boxes when there's some sort of decision-making faculty. So I think it is good that we do think in terms of interpretability. So are these good black boxes? Are they rigorously tested as well in different settings? So I think that sort of understanding is important. But then again, we work with black boxes every single day. My mobile phone is a black box in some ways. And there are a lot of decision-making algorithms that are black boxes that we interact with on a daily basis. But I think that doesn't mean that it's okay. We need to know why decisions are made as well. So it's really important that that as a field in machine learning, we do think about doing AI and doing machine learning responsibly, that the sort of decisions for the end user aren't in any way going to put anybody at risk. And this is really important, especially in fields like healthcare and in other fields as well. There have been so many examples in the news where we see what the potential risks are as well. But at the same time, it's an opportunity for us to scale solutions and to have solutions being deployed rapidly as well. And this is really important. AI and machine learning, they're powerful tools for different applications in healthcare and immunology as well. But I think that thinking of this nuance of who is the end user and how do decisions affect the end user, are the models fair, are they transparent, and are they interpretable as well? So there may be moments when you do need to have that level of transparency. For example, if a decision is put into question, I really believe in having systems, machine learning systems, where there's a human in the loop as well, because you don't necessarily want to delegate all of the decisions to a machine. Sometimes you need a human. So it's all this to say it's very particular on the use case, and we really need to think carefully of what are the use cases and what are the scenarios as well. But yeah, I think it's very important. question. But also, when we think of black boxes, very often I also think in the fields of machine learning as well, that we need to know what are the mathematical underpinnings of models as well, because it's not good enough for us as a machine learning community to say, yes, deep learning works, but we do need to understand how do things work in order to be able to train algorithms in a more efficient and better way as well to reduce any sort of error and bias.
Yeah, I like those notions, and especially the training these models in a way that is efficient is super important, but also in a way that is respectful of the end user and that the application does not harm people in the end. Those are super important notions. Thank you so much for bringing these up. I'll switch gear a little bit, and we'll talk about your work in particular. Can you both describe your journey in integrating AI with immunology? Because this is what we're all excited about, and so in particular in your work, how did you came to it, and what do you do today integrating AI in immunology? Bryan, you want to start?
Sure. So, I often, a lot of my research program focuses on host pathogen interactions, specifically BL3, slow biosafety level 3, slow growing pathogens. And so I always advocate, like, look, I would love to be able to operate a little bit faster where I can. And oftentimes I think about machine learning models helping accelerate either decision making on experimental design, deriving insights from what other people observe in other infections, trying to better accelerate. Something we're working on really heavily right now is using machine learning to accelerate TB vaccine design. And so I always kind of thought of this as an urgent need that I really needed to have as part of this my research program, as opposed to ever being what am I trying to say here? I took like linear algebra and a machine learning class as a graduate student, but it wasn't necessarily something that I was going to focus my entire research program on. It was something that I wanted to have a fluency in and wanted to be able to deploy as a tool when it felt appropriate. And it just so happens that I think in my worldview that there are many questions in host pathogen interactions, specifically in the setting of tuberculosis, where machine learning can be really, really helpful. And so over the past few years, we've tried to work on different questions there. So thinking about how we can better accelerate the identification of small molecules that potentiate bacterial control inside of immune cells. Can we better understand how to compare the immune cell landscape that people are observing in tuberculosis, which obviously I always say, like, this is particularly important to me because, working in TB, we know we're kind of a small community, and we know that the research funding is a little bit distributed in a different way for different folks, right? And so TB doesn't have all the resources that cancer does, for example. So I would like to be able to utilize our TB data sets and see what people are seeing in cancer and borrow from their observations that might then help, at the end of the day, accelerate what we're able to do in TB. Because, hey, if you figured out that this cell population is detrimental, and if you can target it with this drug over here, and I see that I have this cell population in TB, hey, thank you so much, cancer field, for helping me identify a potential intervention without having to spend the 25 years that it probably took them to get to that moment. And the last place where we're really thinking about this a lot is trying to come up with new ways to predict what antigens are relevant for TB vaccines. And this is a really new area for us based off of, just thinking about human complexity and T cell responses. And so if we think about the complexity of T cell responses in humans, We have diverse major histocompatibility complex alleles. They each have their own kind of peptide binding preference. For model antigens, this is easily studied, but for TB that is antigenically complex with 4,000 proteins, we've got complexity on complexity on complexity, but we would ideally like to make vaccines that work homogeneously across the human population. So we've been really thinking about how do we use machine learning to better accelerate figuring out which antigens are relevant and will work across human diversity. So I'll stop there.
Excellent. Thank you so much for telling us about how you came to it and what you're doing today. Danielle, what about you?
Okay, so when you first asked this question, I thought, oh my gosh, I'm a bit of an imposter here, because I don't actually work in immunology, but actually I do in a way. So I think you can say my journey into AI for immunology is from a more clinical perspective, and trying to understand underlying mechanisms. And in order to understand underlying mechanisms, you need to do collaborations with immunologists as well. So my background, as I said before, is in mathematics and statistics. My PhD, it was looking at the limitations of statistics in observational birth cohorts. So looking at longitudinal data to identify, can we understand heterogeneity in asthma and allergic diseases? So this was in the early 2010s when there was this hypothesis that drugs are not one size fit all, and the expression precision medicine or personalized interventions came in. So my PhD and a lot of my research has been around identifying subtypes of disease that very often terms like asthma, umbrella terms for multiple different conditions. And by understanding what those different conditions are on a population level, then we can identify personalized interventions as well. So I published several papers in that field. For example, we identified 5 different subtypes of asthma, and we're able to identify a subtype that didn't respond to asthma medication. And this was just 3% of the population of asthmatics who didn't respond. And backstory, about 6 years after I finished my PhD, I went to GlaxoSmithKline for the first time. And at that time, there was a drug on the market that was targeted for IL-5 asthmatics. So I think it's really interesting understanding those mechanisms. But a lot of this research that I did in asthma and allergic disease in particular, we did collaborate a lot with immunologists and basic scientists to understand what are the underlying mechanisms, what are the causal mechanisms of those different subtypes of disease. Because by understanding those mechanisms, then maybe we can get more targeted responses. So I'd say that was my first touchstone of how I got into immunology. We could think of causality and causal reasoning based on the data we have at hand, but sometimes you need to generate more targeted data based on hypotheses. So these hypotheses of what sort of response, at that time we were looking at response to RSV and different viruses and bacteria as well, of PBMCs. And at this time, I think it's really important, once you have a hypothesis based on the data, so based on certain genetic responses, then we could look at different targets to understand those mechanisms of disease as well. So that was my part one of getting into immunology. Later, I worked in other things that did not have to do with immunology. And now I'm at GlaxoSmithKline. So at GlaxoSmithKline, I think it's, at GSK, it's an exciting time. So I had a series of teams there looking at the end-to-end pipeline of drug development and of pharmaceuticals. So there's one VP who looks at, I would say, more of the immunology side. So looking at the basic building blocks of life. So looking at RNAs and looking at how we can do drug discovery using, by understanding mechanisms in RNA sequencing. And on my side, I'm looking more at the clinical aspect. So more at phase 3 and 4 trials. But at the same time, when you're looking at heterogeneity and responses to diseases, so a couple of the conditions in my portfolio. One is hepatitis B, and the other one is looking at oncology as well, so different cancers as well. And I think in this area, it's really important when you're understanding what are the subtypes of responders. So we are able there, again, to identify subtypes of responders and non-responders to different drugs on clinical trials and in the market as well, to get more targeted responses and better responses for patients. So getting the right treatment to the right patient. At the same time, it's important to understand those mechanisms. So we do a lot of work on drug discovery and also understanding, for example, cancer cell targets as well. So why are people responding? So I think that this is a really important field in understanding what are the building blocks, what are the targets as well, so that we can have more effective interventions. And I think for me, it's a really exciting field. So I have straddles between academia and industry as well. I spent some time working in tech, in DeepMind and Microsoft. And I think for me, the number one thing of being able to work in pharma is being able to have impact on so many patients as well, and being able to have that influence in how we think of drug development as well. So getting interventions to different subgroups of people. So I think this is a very exciting field to work in, machine learning and AI for health.
Absolutely, I agree. I think it's a great time and a really exciting moment in history where we're going to be able to use those tools. Can you actually share? Maybe it's early, but can you actually share a success story where you've been able, or your teams, or some other teams have been able to stratify groups of responders and non-responders thanks to AI, and come up with a drug that would work better? Are we there yet, or do you have an example?
So coming up with I think one of the things you were just saying, that it is an exciting time. I think one of the reasons why it's such an exciting time is the scale of the data that we have as well, that we're able to generate biological data at scale. This is something that wasn't in existence 10 years ago, or maybe 15 years ago, or also we didn't have the computational power as well. So I think the marrying of the two, the computational power to build machine learning algorithms that are fast and that scale as well, but also the scale of data as well it's been a pivotal moment in science, and I think in AI for life sciences and for healthcare as well. So I alluded to one particular example, which was around liver disease. So looking at hepatitis B. So there is a drug in clinical trial at the moment at GSK called bepirovirsen. And we were able to look at the phase 3 trial data and identify a subgroup of patients, that different subgroups of patients based on their omics data profiles. So we identified 5 subgroups, and we were able to see those who we should target for interventions and those who maybe would respond to something else. So there are these sorts of success stories where it's not just, I think it's not just a data-driven approach, but it's also biologically plausible. So there are mechanisms for understanding why the data is telling us that, and also that we could identify those subgroups based on response to treatment as well. So yeah, I think this is quite exciting as well.
Yeah, that's a fantastic example. I'm glad to hear that we're already here, right? That we're already using AI ML to distinguish groups of patients, and that we can help find the right treatment for them. That's amazing. Coming back to you, Bryan, you mentioned sorting through the molecular complexity of those large families of molecules. Do you have a good example or success story of you've been able to use AI approaches to really pinpoint the molecules that were important, or the variants that were important?
Yeah. So 2 quick examples. So early during the pandemic, we actually established this machine learning framework that allows us to, in an unsupervised manner, make predictions about viral variants that would be more likely to escape neutralizing antibodies using a computational framework that involves protein language models. But we also, at the same time, we're actually using protein language models to do drug discovery. So trying to take a protein language model's representation of a protein sequence and then pair that with a computational representation of a small molecule in the same way that we think about these matching algorithms that try to match, something with something else. We were thinking about how do we do matching algorithms that match a protein with a small molecule that actually may bind it. And so the idea was that could we screen an 11,000 molecule library in silico instead of having to do that in the biosafety level 3, because without a liquid handling robot, we know how that story would end. And so we actually did that exercise, and what we found is that, yes, we were able to make predictions, but the dirty secret, I will say, just to spice it up a little bit, is that biological machine learning is hard. Biological machine learning is hard because oftentimes we don't have a lot of examples from which to draw from. I know that we as scientists feel like we're doing the most, but these machine learning algorithms oftentimes really want a lot more data than we can offer. And so one of the things we also learned in that exercise of doing the small molecule discovery exercise was that we really needed to understand where we were uncertain about a particular prediction. And that was actually really foundational in how we thought about improving our algorithms and improving our drug selections, is that we had to have an estimate for how confident the model felt it was about the prediction it was making. And I think this is going to be a frontier that we have to confront in the biological machine learning space, because a lot of the algorithms that are being developed that people utilize are for like large high throughput data sets that are data sets like of the Google and the Netflix kind of scale. And how do we scale that down to something that's of the scale of what biologists are actually doing, which is still not like, just think about how many people watch Love is Blind on Netflix a day compared to the number of people in the world doing the exact same experiment you are in lab.
Right. And sharing the data, right? Because if we accumulate all those, sequencing runs from multiple places around the world, we might get to those numbers, but we're not there yet at sharing this amount of data so we can actually use it. And also there's all this batch correction that comes in play. But yeah, exactly. I agree. I think we still have some way to go to use those tools with the amount of data that would make those tools the most powerful. I like how you've been thinking deeply out to define the limits of these predictions that you can make with these tools. That is super important. It's a good segue into my next question, that is, what are the particular challenges that you've encountered? And Bryan, you just mentioned a couple here. So if you have one more, feel free to add, but I'll start with Danielle. Is there particular challenges that you've run into? And maybe it's the same kind of challenges with the amount of data and this sort of issues.
Yeah, so I think data is a big challenge. Getting data at scale. I think also there's the reverse challenge as well of developing methodologies that scale well in sparse data settings. So this is actually an active area of research that I think is really important in this field, where you have where you're trying to understand data sparsity. How can you model data sparsity and understand mechanisms of data sparsity as well, mechanisms of missing data, whether it's on the clinical or immunological side. These are active areas of research where we haven't gotten I think there are some successes, but the successes tend to be outside of the life sciences in doing machine learning on small scale data. I think another challenge is all around the understanding the utility of the model. So one thing is you can create a really good model, a really robust model that works well, that does fantastic predictions. But I think there are two problems. So one, I think the challenge very often is around utility. So for example, when there are lots of success stories around drug discovery and identifying targets. It takes a while to get a drug into the market as well. But at the same time, it is a success story. The challenge is a bit of a success because using machine learning, we can accelerate that process as well by shortening the time to target identification. So that's something that shortens the process by about 5 years to 7 years. But I think another challenge around AI for life sciences, immunology, health is if you build, so very often it's not just about discovery, it's also about how do you deploy the model and how do you use the model as well. And I think as a community, as an AI and immunology community and AI and health community, we do need to do a lot more work around utility and usability as well, and around how to build good products so that people can use these systems and that they're benefiting society as well. So I think for me, one of the biggest problems is translating from a good model and good scientific discovery to clinical impact. How does that reach the patient?
Yeah, thank you so much for sharing those thoughts. Bryan, do you have an extra challenge that you want to share about using AI ML in immunology?
No, I think something I just want to underscore something that Danielle just said is like making it the usability and making sure that it gets in the hands of people who will benefit from the development of the tool. I just like that is so important, right? People have you have to do it in a way that builds trust. I think that's the other thing, that we still have a hurdle to clear when it comes to, especially in biology, is like, okay, cool, you trying to tell me that the computer is smarter than me about biochemistry, right? So I think building trust and building credibility and human in the loop and multiple humans in the loop would be great.
Yeah, absolutely. And I think that connects to the ethical considerations using AI and ML that we've heard, in the large media, especially considering ethnicities and how machine learning approaches could be biased for some particular applications. Is there any way that you're thinking of this in your own work?
Yes. Okay, so I'll just use the example of antigen prediction, right? MHC alleles are incredibly globally diverse. And we know that different people, different continents, different subcontinents have different MHC alleles. And oftentimes we have really great models that work for a particular allele, a particular genotype, but doesn't generalize. And the other thing that's really important is that when we build models, like understanding, how do our models generalize? Is this a model that's going to work for the entire population? What training data did you use? Did you only use training data from a particular population, right? What we learn in adults may not always translate to what we learn in children. What we learn in one genotype, it may not translate to another genotype. And that is really important. And so I always ask, okay, great, you built this model. What was the training data? And I think we need to keep that as really part of the conversation.
Absolutely. Using diverse data to reflect diverse populations, right? Yeah, absolutely. Is this also something that you're thinking of, Danielle?
Yes, I think I totally agree with what Brian said, that uh, it's really important to have diverse data sets, that data sets are representative of the population as well, and the populations that are targeted for interventions. I think there's also something to be said about how do you build this trust, and I think maybe this is a little bit left field, but very often we can think, when we think of things like marginalization, I think also that there is an extent of tech marginalization as well, that maybe very often we don't necessarily engage with as a community, speaking in a Black and immunology community. Maybe very often we don't necessarily engage with technology as in actually trying to build technology solutions as well. So this can be, it may be part of why there is that lack of trust as well. And there's trust on different levels. There's trust on the level of giving data as well, so that our data is represented as well. I think this is a baseline problem. There's a lot of mistrust in different aspects. But I think also if we engage in being more people who want to build solutions, and I think one way of doing that is through educational programs as well, where we build these communities of trust and people, so that trust isn't an issue. It's second nature, that we are not just engaging with technology as consumers of technology, but we are also deploying solutions. And I think this is something that you do see in different parts of the world. When a few years ago I was lecturing in the first deep learning endeavor, which is it's all about increasing participation of machine learning in Africa. And one of the things that really struck me the first time going to Africa was seeing there how people solve problems and how people engage with tech in order to solve problems. I think that this is super important, that we do this as something that is natural for us as well. And in that way, we build communities of trust where we're not just engaging with tech, but also using tech to build solutions as well. It should be second nature for us. So yeah, I think that's not all of the solution, but I think it's something that we can do. So one thing I am really passionate about is education and teaching children from underrepresented groups, and very often these are underrepresented groups in our data sets as well, how to code. It's one angle to tackle this from, but I do agree that that there is this trust that, and yeah, we have to look not just at how we can intervene, but also at the moment of deployment as well, that we make sure we're in those conversations as well to make sure that models are responsibly deployed as well. So I guess it's quite fortunate that I get to do this on a day-to-day basis, that I think about. In GSK, we have a whole team on responsible AI, and these are really important questions to think about, how we interrogate our models and how we make sure they're robust.
Thank you so much for bringing all these points. I mean, you've covered such a broad range of those important questions and issues, and I love the multiple proposals that you've done on how to try to solve this and to get to a better place of trust and natural use of these tools. Yeah, I like that a lot. Thank you so much. I will now maybe see if there's some questions. I see that, yes, Medina is asking the question. For people that do not have statistics or computer science backgrounds, what is your advice on engaging with AI ML? And I'm assuming that it's for scientists, immunologists, but maybe broader than that as well.
Yeah, so just to answer this quickly, I think this is a good question, and it also is a nice segue into what I was saying before as well. I think just engaging with resources, there are lots of resources on YouTube, and also there are lots of online courses that are free. I think Python notebooks using Jupyter notebooks is a really good entry point as well. And the barrier to engaging with AI and machine learning is not high at all. I think that these sorts of toy examples that we get on YouTube, and on YouTube, there are so many things around different applications as well. So sometimes the examples used can be really far away from the questions we have, but you can just look up things like machine learning in for science, machine learning for health as well. And these are really good entry points. And I think also learning how to code, it's not hard. It can be hard at the beginning, but it's also very
steep learning curve, right? But
uh
then once you're above the curve, it's going, right?
yeah, but I think it gives you a different way of thinking and also a different way of interacting with data as well. And this is good training. So just like me as a machine learning researcher, I have no idea about biology or sciences or health. And I think that as a practitioner in the field, as a machine learning person in the field of health, I need to understand the domain. So in the same way, I think that it's great when my colleagues who are doctors or scientists as well do learn about machine learning so that we can bridge the gap and have those conversations. So I think that this is important. There are lots of things on Medium and lots of articles that I think it's good to engage with so that we can have interdisciplinary conversations as well.
Excellent. Bryan?
Yeah, I echo all of that. I also think that if I could suggest one more additional class to think about, I'm really interested in like linear algebra. I just find that a lot of the deep learning and like a lot of the machine learning tools have like a origin in linear algebra. And so I want to just make a vote for that one as well. And there's a great an MIT linear algebra class online for free.
Excellent. I'll have to look it up and to find it. All right. So another question from Zinga Mac. Do you have any suggestions as to how I can get an overview of what the various tools are and how they can be used in biology? I see a lot of standalone several month courses of AI and ML in business on one end, and AI ML master's degree on the other end. Any suggestions for something in the middle? I think it talks to a little bit what you've said, Danielle, right? That there is some resources, YouTube video and tutorials that can be, very simple and not necessarily a whole course, right? It feels like there's different degrees of resources on YouTube in particular. I've seen that too.
Yeah, I will do a plug for also like I understand the thing of time as well. So there's a book by Eric Topol called Deep Medicine, and I think that book is quite good because it's quite high level. It gives quite a high level overview of machine learning / deep learning for health. It's very well written as well, very engaging as a book. So I would recommend that book as well, just to get an overview. And it's good for starting a conversation and looking at what are the motivations for looking at AI in healthcare. The other thing, as Bryan was saying, MIT, there is actually a course from MIT by David Sontag's group, professor. I think he's a professor at MIT. I think he is a professor at MIT, and he has a course on machine learning for healthcare as well, which is super comprehensive. It also covers areas of fairness as well. It's more geared towards clinical data, but it's very, very, very good and very engaging.
Yeah, I'll make one plug for something. A
I don't know
nature
if you
review.
can hear me. I have lost the sound. So
Oh, no worries.
We can hear
I'm
you.
trying to figure out how to recover. I guess I'll ask the last question. You can hear me, right?
Yep.
Thumb up if you can hear me. Yes. Okay, excellent. So I'll ask the last question, that is, where do you see the field of AI in immunology heading in the next 5 to 10 years, and what the breakthrough do you anticipate?
So I will give what I'm really looking forward to, which I think would be transformative, is this idea that I think the protein structure prediction revolution is really remarkable. And I think two things that I'm really excited about and nested in that is protein structure to drug target engagement, finding small molecules that bind a particular structure, and we can even predict the structure of the small molecule bound to the target. And then the other place is really thinking about how we predict structures of components of the adaptive immune response cascade, be that predicting antibodies, predicting peptide MHC structures, or predicting T cell receptors. Those I will have to do I'm waiting to do my sabbatical to find the lab that's going to like drop the new best thing, and they better be ready for me to come visit for a year.
That sounds great. I don't know if Joel has sound, but probably not. But I am all for that. I'd love to see that in 5 to 10 years. I think that the thing I'm excited about is how we really push the boundaries of machine learning for personalized healthcare and really get personalized and targeted interventions for patients as well. So, and I think this links also with how we look at the drug development pipeline as well. So I'm really looking forward to that in the future.
Well, I still don't hear. I'm sure that those answers were fantastic. I'm going to take the cue that this means that we are going to close the session. If I couldn't hear the last responses, all the session was fantastic. Thank you so much, Bryan and Danielle, for answering all those questions and really giving you a broad sense of how AI and ML has been using currently in immunology, and also what the big technical and ethical questions are, how to approach all this. Thank you so much. And I'll close the session by just mentioning to our audience that we have a closing keynote session that is following this session. So stick around. We'll probably do a 5-minute break. We're running a little bit late. But we'll have Dr. Glenn Simons to close Black in Immuno Week 2023. Thank you so much.
Okay, thank you. Nice to meet you both. Nice to meet you, Bryan.
Likewise.
All right, take care. Bye. Thanks, Joel, even though he can't hear.